shared fax facility instruments. This is the old one, Fax 2, used for a lot of multicolor immunofluorescence with a dye laser system. Here we have the FaxStar Plus cell sorter, a relatively stock commercial system that we use also for two laser with visible and ultraviolet illumination. Analytical room, computer terminals, and Macintosh, things like that for manipulating graphics. The third routinely used instrument here, a fax scan with custom software that we'll look at later. Okay. Go. And around here in the next room, a new instrument under construction to have essentially to use all the capabilities of the other three in a single instrument with our own custom electronics and computer system.
Okay, good. Oh. I hadn't even thought. Yeah. No, I think we're here in mindset here. Let's do this.
Yeah, that's good.
So we'll work our way back there.
You rolling?
Okay, rolling.
Bye.
Back to speed. And speed.
This Friday morning, February 1, 1991, we are at Stanford University in the Beckman Building, Department of Genetics, looking at the fax shared facility, one of its laboratory rooms. Our guide is David Parks, director of the fax shared facility. David, could you tell us what we're looking at in this room?
Okay, well this is our two laser cell sorter using two colors of visible illumination. It's essentially a descendant of the one that's over at the museum now, in that the bench part that we're looking at now is an update on that system. Electronics, see later, is even more closely related. We could go in and this system uses excitation with two lasers. One, which you can see a little of here, of blue, typical, and turn on the dye laser in yellow-orange. And this is used to excite frequently 4 different colors of immunofluorescence, 4 different reagent measurements on each cell. So, although this is now quite an old system, it's still doing state-of-the-art research.
So the original BD2 had one laser, and now another laser was added to this system.
Right. This is has a second laser, has 2 argon lasers, one of which is used as the power source for a pump dye laser.
Okay. And about the electronics system, you say is more or less the same?
Yeah. Looking over at the electronics, this is probably the last of the Fax 2 type electronics that was built, and we got it because it was more flexible than the what was considered more modern versions at the time we got it, and allows us to do things. For instance, this originally came with only 2 sets of sorting windows, 3 measurements intended. We expanded it out to 6 by building more electronics ourselves. Okay. The system communicates with the VAX computer systems here through the interfaces, as seen down here is the technical. Some of the changes that we've done on it is, see, we've sort of rebuilt a lot of the system, including displays to be able to handle the 6 measurements and all. And also, up in here is the automatic cloning device, which was an early development here, but we still use the old external cloning device, which has been internalized in the newer models.
And the machines on the rack.
Then finally, for here, a rack of high voltage power supplies and amplifiers that are computer controlled, so that now the instrument standardization has been taken over by the computer. This instrument was originally not designed to have any sort of computer control, and we've added that partially at this point. So it makes it possible to standardize the instrument and have the computer know what the instrument conditions are, because it sets them.
And more of these instruments that you added have now been incorporated into the VACStar Plus that we will see in the next room?
Yes, it's partly because as a result of work that we've done, and partly as a result of just the way things go, and we've retrofitted this old instrument, kept it running to do more modern things.
Good. Cut.
Okay, and speed?
Okay, and yes, down in this corner is the interface, which transfers the measurement data on the cells to the main computer system.
Okay.
Okay, cut. Good. One whole cut. One whole. And live speed. And speed.
We're in the second room of the VAX shared facility. What do we see in this room, David?
Okay, well, this is a VACStar Plus. So pretty much a current instrument like you've seen at Becton Dickinson. It runs visible laser and a UV, so it is a 2-laser system for a different set of measurements than the previous one. Overall, it's basically a stock instrument, except that it does have connection to our own computer system and software.
Okay, let's cut.
Rolling. Stand by. Speed.
Okay, so here we're looking into the real business area of the cell sorter. The greenish glowing object is the nozzle tip that the cells and the jet of liquid are coming out of. The laser beam intersects that jet just outside the nozzle. We take the measurements from the light signals generated at that point, and just a little further down, the stream is broken up into small droplets, which, by charging them and passing them through a high voltage field, results in moving the drops, deflecting them out of the central stream to yield sorted fractions, shown here, where we have two different criteria on the measurements, which result in left sorts and right sorts into 2 different collection vessels.
Okay, can we keep rolling and tilt back up and get a static on that intermediate, that window there.
We're at the aspirator level here, the undeflected drops, those that do not contain cells of interest and are to be discarded or collected immediately, allowing the deflected drops containing interesting cells to go past and on either the left or right side and be collected.
Okay, pause it for a second.
Quiet. And back to speed. And speed.
We're in the third room now of the fax facility. What are we looking at in this room, David?
This is a Becton Dickinson fax scan instrument. It does single laser analysis. It's not a sorter, but it does have the capability of doing 3 colors of fluorescence if you have the right reagents to do it. So it's quite a capable instrument and generates quite good data. So we've taken this as a relatively stock basic instrument and connected it over to our own computer system. Here you're in the looking in at the business area there. The flow cell illuminated by the blue laser light, where the laser comes in from the left. The cells are flowing upward through the flow cell, and the fluorescent light is collected by a large immersion objective in the back, so it's very efficient at collecting the light, even though the laser power is fairly low. And then the light scatter detector is off to the right. So this gives us a total of 5 real measurements on each cell.
Good. And we're looking at the computer terminal.
Right. This is the prototype software on workstation, where it's running some test particles. Actually, 2 dot displays here of different views of the test particle data. We have also an instrument control console for setting up the instrument conditions and controlling the different parts of the instruments, amplifiers and things like that. And then another window in which the instrument calibration is done and standardization. Here's just a running display of the measurements on each of the 5 primary measurement channels plus 2 pulse processed channels for pulse width and pulse area of one of the measurements.
How is this program different from the standard one that goes with a fax scan?
Well, it's totally different. It's one that has been generated from basic concept to this implementation entirely here at Stanford, and pretty much completely independent of the Becton Dickinson software. So we're not trying explicitly to compete with them, but we are working as an academic development environment here, and using the fax facility customers as our guinea pigs.
And what are the major advantages as you see this, or as a scientist see it from this software?
Well, historically, the software that we've developed, the fax desk software is called, is particularly good as a data management system, that we really have access right now to all the data that's been taken since 1983 on this system. And so that archive capability, the fact that it runs complete list mode data so that you can go back and effectively reconstruct an experiment, reanalyze the data years after it was originally taken. Okay. Important. A number of other factors in terms of the data display and all were unique at the time they were done several years ago in that part of the system. The idea involving this system console is new in that it's working in basically quite artificial intelligence, but it's a rule-based control system. So the methods that are used by the computer to understand what we're doing in controlling the instrument are quite different than in the other software systems, even though you could generate that actual display in other ways. The way it goes about deciding what you're telling it to do and everything is fundamentally different than the sort of explicitly programmed types of software.
Good.
Okay. Keep it rolling. Keep it rolling. Okay, I think we've got what we need here. Waho,
Yeah.
pause tape. And just re-identify this, just a couple of words, what we're looking at here.
Okay, so here we're
Speed.
Okay, just one or two words. This is the,
here we're seeing the flow cell area of the fact scan illuminated primarily by the light from the laser.
okay, good. Cut.
Rolling. Hold for speed. Speed. And speed.
We're now in the fourth and last room of the fax facility, looking into the future, I think, Dave, right?
Yeah, this is our new instrument development area. What we see here is the electronics rack for a new version of cell sorter that we're in the process of assembling. The optical bench, lasers, that sort of thing is being built onto a stock Becton Dickinson FacStar Plus bench, but we're expanding it out to have visible ultraviolet and dye laser illumination, and thereby be able to take any of the measurements that are available on any of the other three instruments on this single instrument and be able to do it in any combination or all of those measurements at one time. So it's intended to be a very versatile and flexible and powerful instrument in terms of its measurement capabilities, but at the same time be tame enough in a sense that biologists, at least our better trained biologists, can run it for themselves without an engineer standing over them all the time. So that's the reason for using the commercial bench as our base. The electronics, however, is entirely designed and built here with what's called pipeline processing in the signal, so that all of the measurements are taken and dealt with as rapidly as possible, so that we would hope to be able to maintain higher rates of cell analysis and potentially also of cell sorting than is possible with the ordinary electronics. The electronic system communicates with a special version of the VAX computers that use, and part of the reason for the existence of this system is to be able to carry out new forms of cell analysis and in terms of computer processing, cluster analysis, finding and analyzing populations of cells, and then be able to transform that automatically and directly into sorting criteria, so that we can actually do an analysis and then carry out very general sorting specifications on a set that may contain 10 or more measurements per cell.
And is this a rule-based system as well?
Well, that's a different part of it. The control system for this, the console and user interaction with the electronics and things, will be based on that same rule-based system that's being prototyped on the back scan. The cell sorting is a different issue, but it is a very open computer geometry, so that we'll be able to do all sorts of anything we want to do in terms of the analytical capabilities, and be able to transform those into sorting criteria in a direct fashion.
Okay, this is room tone in the pattern sorter. Room tone.
We need long ones.
We have seen some views of the Herzenberg laboratory at Stanford University. Now we are speaking with Dr. Leonard Herzenberg, professor of genetics. Dr. Herzenberg, we'd like to explore some of the research work that is being done with the cell sorters. Maybe you can start with some examples of the early work that was done and bring us up to the present.
Well, in the few minutes that I think are available for this, we'll be covering more than about 25 years worth of work of quite a large number of people, many of them working in this lab and also a lot of them working around in other laboratories at Stanford. The original reason for developing a cell sorter, as I told you, was to try to test the clonal selection theory, that is to try to isolate antigen binding cells by labeling these cells with fluorescein or another fluorochrome and sorting with the sorter. We were able to do this, and we were able to prove that the cells that bound a particular antigen sorted with the sorter would then make antibodies against that particular antigen, not against other antigens. So this was a formal proof of the clonal selection theory of antibody formation. In the years since, there's been many studies of cell surface molecules, cell surface antigens of cells that are active in the immune system. These are both mouse cells and human cells. And because of the development of hybridomas, which make monoclonal antibodies, and our application of monoclonal antibodies to identifying cell surface molecules, and many other people who have participated in that effort as well, and many people have been able to discover dozens, or in fact at this point it's probably at least 200 different antigens, which are different molecules, therefore, which are expressed on the surface of different kinds of cells in the lymphoid system, in the antibody-forming system, in the blood-forming systems, the hematopoietic system. And this has made it possible to classify these cells into different types, to study their development, to describe the lineages that occur in development. Our own work focused for a while on regulation of the immune response. We isolated with the sorters the cells which were suppressor cells, cells which were antibody-forming cell precursors, T cells which were helper cells, and saw how they interacted. That was a major area of activity for us at one point. We then moved into some more molecular kinds of studies where we were able to use the sorter, the FACS, to help us to clone genes. So we transfected, transferred DNA from cells of one species, usually mouse, usually human, into recipient cells, which were usually mouse. And the expression of the human genes for cell surface antigens could be detected on the surface of the mouse cells, so the human antigens expressed on the surface of the mouse cells, by staining with fluorescein or other fluorochrome conjugated monoclonal antibodies, and then using the FACS to sort out these cells, and by a complicated process, but not so uncommon in molecular biology and gene cloning, we were able to clone the genes for several of the basically important cell surface molecules. And some of these are now very important in clinical work. And that could bring us to the current day, but there were a number of other steps along the way, which perhaps I should tell you about first. So we'll come back to AIDS, where some of the work on the cell surface antigens led. My first postdoctoral fellowship was at the Pasteur Institute, as I said previously, working with Jacques Monod, and I worked on the enzyme beta-galactosidase, regulation of that enzyme in bacteria, where beta-galactosidase of that sort was made. Then some years later, a fluorogenic assay for beta-galactosidase was developed, and then just a few years ago, in 1986, I chose to go on sabbatical to the Pasteur Institute to do some more work using that particular enzyme, beta-galactosidase, from E. coli, coded for by the gene LAC-Z, to see if galactosidase could serve as a reporter molecule for gene expression, for RNA transcription, and for other purposes, where we would be able to identify or quantify the amount of transcription in individual cells in a population, rather than looking at, as is conventionally done, an extract of large numbers of cells and just measuring the amount of activity in that extract, which has the biggest disadvantage from our point of view of not giving you any information about the distribution of the amount of gene transcription as measured by the amount of beta-galactosidase in individual cells in the population, but rather just gives you an average number, and that average could be made up of some cells that are making no enzyme and therefore have no transcription, other cells which might have a little bit, others which have more, and maybe just a few cells that have a very large amount, or a large number of cells which have a large amount. None of those different situations could be distinguished by the extracted method, but they could be distinguished by looking at individual cells. And the FACS was perfectly designed for that, and the substrate that we could use with beta-galactosidase was also perfectly designed for that. I mean, it was a fluorogenic substrate, fluorescein digalactoside, which, as I said, had been used previously for other purposes. We learned how to get that substrate into cells. If there was beta-galactosidase in those cells, they would hydrolyze the fluorescein digalactoside to fluorescein. Fluorescein is fluorescent. I opened it to say that the substrate, fluorescein digalactoside, is not fluorescent, but upon cleavage of the galactose residues by the action of the enzyme, you generate fluorescein, which is fluorescent. So we learned how to get the substrate in, and then we had to solve the problem of how to keep the product from flowing out, how to keep fluorescein from flowing out, which it normally does. We found that just by lowering the temperature below about 15 degrees centigrade, and we normally work at 4 degrees centigrade in an ice bath, the fluorescein doesn't come out. It'll stay in cells indefinitely. So then we can pass the cells through the FACS and measure the amount of fluorescein in the individual cells as they pass through the FACS. Now that also, so we made that work while we were on sabbatical. Some of the work was out at Stanford, and students who have recently left the lab, Gary Nolan and Steve Firing, were much involved in that work. And now, a few years later, we're using that technique or that reporter technique, we'll call it, to do a number of the projects that are going on in the lab now. One of them involves the AIDS project, which I alluded to earlier. AIDS, of course, is acquired immunodeficiency syndrome. It's caused by infection with the virus HIV, and is a real scourge to mankind now. And in the United States at the current time, this is 1991, there are reported to be well over 1 million people who are infected with the HIV virus, and under current situations are expected sooner or later to die of that viral infection, manifestations of the viral infection, unless we can find some way to delay that event indefinitely, or actually cure people of the virus, or cure them, certainly the effects of the virus. So a couple of years ago, we did start to look at regulation of that virus using the beta-galactosidase reporter gene system, and found that, and this is not work of our lab alone, but of many other people, and just because of the brevity of time available, I'll try to summarize it, that the virus HIV infects cells and is often found to be there in latent form, that is not actually producing virus. In other cells, you can infect the cells acutely, and the virus will be produced, and the cells will die. In other situations, the virus can be produced without the cell dying. But in any event, the study of the virus in our lab started by using the promoter of the virus, called the LTR, long terminal repeat of HIV, ligated to or attached to the beta-galactosidase gene. the coding sequence of the gene. That then allowed for a measure of how much viral transcription was going on in these cells. So we measured viral transcription, and if we have time, one of my students will talk about an assay we're trying to develop, a very rapid assay, where there is no rapid assay now, for viral production. So we've used the FACS to both isolate the cells, which have constructs in them, and to measure the rate of viral transcription and viral replication in cells using this beta-galactosidase reporter gene technique. Now, you want me to talk further about the work going on here?
Well, I think how are we doing for time, Brian?
Okay, we have about 40 seconds.
Okay, let's
Oh,
cut
boy.
this in.
Okay, so what do you want to do?
Now, I think
it's
that would be
hard
a
to-
great introduction to, did you want one of the people
Monty.
who was working on that assay to say a few?
Yeah, if you could.
Quiet. Speed.
Okay, go ahead.
Okay, I'd like to introduce Monty Montano, a graduate student in the genetics department in this lab, who's going to tell you of one interesting and I think very important application of the reporter gene technique using beta-galactosidase and the substrate fluorescein digalactoside, FDG, and of course the FACS to assay for it. Monty?
Thank you, Len. As he mentioned earlier, we have been working on developing an assay for detecting infectious HIV virion. Basically, our strategy was to transfect a reporter gene, the HIV LTR attached to LAC-Z, into a cell line and select for a stable clone. Once obtaining this clone, which did not express CD4, the HIV receptor, we transfected a CD4 expression vector into the cell line and then again selected for a CD4 expressing clone. Now, we tested whether or not this clone could be infected with HIV, both laboratory strains and patient strains. And in fact, the infection does result in the activation of this reporter gene. Basically, beta-galactosidase expression is increased, and with the hypotonic load of the fluorescent substrate FDG, we have a FACS selectable and quantifiable marker. Essentially, we can quantitate the number of viral particles and their infectious activity using the FACS machine. An important aspect and utility of this technique is that you can monitor HIV infection in patients by isolating their PBMCs and titering for their virus. Furthermore, because this system can be adopted for viable cell genetics, we can study the infection in viable living cells. Therefore, after our reporter cells are infected, we can measure various parameters in the same cell during the course of infection, thereby allowing us to investigate a concerted cell biological phenomenon in the virus as it undergoes its life cycle. And furthermore, it's important to develop a rapid assay in order to measure effectively the antiviral drug efficacy for patient therapy. And because the system is relatively rapid, and it hasn't been perfected yet, but the goal is to develop it in such a way so as to use it with clinical monitoring of patients and drug therapy, so that we have a readout of the efficacy within the same week as the therapy is being undertaken.
Well, thanks, Monty.
Thank you.
Quiet. Hold for speed. And speed.
Okay, go ahead.
I'm going to introduce now a colleague from another department here in the Beckman Center at Stanford University. It's Dr. Mark Krasnow, who has worked with the assay which I talked about earlier, using use of beta-galactosidase as a gene. But he's going to use it in a very interesting and novel application, which I'm personally very excited about, and I think it's going to be extremely useful for many studies in, we'll call it developmental biology, and perhaps genetics as well. So, Mark, could you Talk about this new idea.
All right. The point, the application of this new LAC-Z reporter gene technology to development was important because very early on in development, there are many different cells, and but those cells look very similar to each other, even though we know from a number of different genetic assays that they're expressing, they are different in that they're expressing very many different gene products. Now, in the last couple years, many people in Drosophila and in other organisms as well have introduced into the animal transgenes in which a particular gene product, LAC-Z, beta-galactosidase, comes under the control of a cell type specific promoter, which allows beta-galactosidase to be expressed in a subset of cells of the very early embryo, and therefore can be distinguished from other cells in the embryo based on their pattern of expression of beta-galactosidase. So with the advent of this technology for being able to identify live cells based on their beta-galactosidase pattern of expression, we could then sort those cells. If we could just take the cells from an early embryo expressing beta-galactosidase, dissociate those cells, and then purify the cells expressing beta-galactosidase and study their development in a test tube in vitro. And I think the real power has come from the large number of such strains which express beta-galactosidase in different cells of the embryo. And so it's then possible, or should be possible in the future, to purify almost any type of cell you want from the early animal and then watch its development or study its properties in vitro when separated from all the other cells of the very early embryo. And this is an example of one such early Drosophila embryo here, shown in this top panel, where the cells, you can see cells in black here that are stained positive for beta-galactosidase, whereas all the other cells of the early embryo do not stain black and appear gray in this photograph. And so it's possible then, using this technology worked out in the Herzenberg lab, to separate these stripes of cells, which are different only in their pattern of beta-galactosidase expression, shown here, and no other differences from the other cells, and then to purify the stripes then away from the interstripes, which don't express beta-galactosidase, in between the sets of cells expressing it, beta-galactosidase. And so then to study the properties of these cells in vitro. And I think eventually this kind of technology will be able to be applied to many different types of animals, including some animals that are even more closely related to humans, like the mouse. And I think the goal eventually will be to not only study, for example, the development of the nervous system in vitro, which we've started to do with these Drosophila embryos where beta-galactosidase is expressed in the progenitors of the nervous system, but also to then begin to reconstruct development of a whole animal from different sets of purified cells and see if we can reconstruct development in a test tube in vitro from mixtures of these purified cells. Put the animal back together again after taking the animal apart, cell by cell, because of this kind of an assay system. This is just the overall scheme here, where we start with an embryo with a LAC-Z transgene expressed in a particular set of cells, and then let it grow to a particular developmental stage, shown here, where there are, say, several hundred cells in the animal, but only a few of the cells are expressing beta-galactosidase. And then we take that animal and dissociate all of its cells just by dowsing it and mechanically dissociating the cells in the test tube, and we get a population of cells, but using the fluorogenic beta-galactosidase substrate, only the ones expressing beta-galactosidase then fluoresce, and then we can purify those cells using fluorescence-activated cell sorter and monitor their development in vitro. And the practical importance right now, or the practical advantage right now, is that there are thousands of different lines which express beta-galactosidase in different cells, not just this subset of cells, but in any set of cells. And so we can obtain from this very early developmental stage, when there are no other apparent differences between cells, we can obtain any kind of cell we want for study in vitro to watch it develop and to study how it interacts with all the other cells in the animal once those are purified.
Well, I appreciate that. It's a really nice method.
Let's do this. Let's start right there with the second one here.
Okay.
Just do the talk down, as I call it, going down. The rest of it is fine
As
with
a-
the wide shot. But he will go close now, and you start it up here and just repeat that.
Repeat that.
Do you want me to hold it in some round? Or
No,
do
he
you
can
have
hold
to
it,
show?
No,
You
I
don't
think
want
it
him
should
to
be.
point at all?
Yes,
I
I
see.
do want him to point. I want him to do exactly what he did before again. We can cut between the two.
Still rolling?
Oh, we'll stop.
Quiet. Speed. And speed. Pause it. Pause it. Rolling. Hold the speed. Speed.
Okay, go ahead, Mark.
This at the top panel shows an early embryo, and what happens during development is that the cells of the embryo divide. The fertilized egg divides many times to give a few thousand cells, shown in the second panel here. Some of those cells begin to express beta-galactosidase, and they're shown in black, as dark black circles, and they express beta-galactosidase because they are within this animal. All the cells contain a LAC-Z transgene, which is under the control of a cell type specific regulatory element, allowing it to be expressed only in a subset of the cells of the embryo. Now, because those cells, although they don't differ in any other properties that we know of from the rest of the cells in the embryo, although they differ in their pattern of beta-galactosidase expression, we can use the fluorogenic beta-galactosidase substrate, which then allows the cells, once the whole cells of the embryo are dissociated, as shown in the next panel, it allows the dissociated beta-galactosidase expressing cells to become fluorescent, and they can then be separated from all the other cells of the animal so that we get a purified population of cells using the fluorescence-activated cell sorter, as shown here. Once these cells are purified, then, we can analyze them directly, test for various properties, or allow them to continue their development in vitro to see if their development then can continue in the absence of all the other cells of the animal. And by doing this type of experiment with many different types of embryos where beta-galactosidase is expressed in many different types of cells, we can then begin to study the development of almost any kind of cell from the early embryo in Drosophila and presumably in other animals as well, although that's yet to be tested.
Okay, very good. Stop tape.
Rolling. Hold the speed. Quiet. Speed.
This is an example of a very early Drosophila embryo in which it harbors a LAC-Z transgene, which is expressed only in a subset of the embryo cells. And you can see after a histochemical staining here that the cells expressing beta-galactosidase appear as black stripes here. They occur in clusters and thus appear as black stripes. And the cells which are not expressing the LAC-Z transgene appear as gray cells. And using the whole animal cell sorting technique that we've developed, that you can then separate the stripes of cells expressing beta-galactosidase away from the interstriped cells which don't express the gene, and then study, therefore, the properties of these cells when apart from the other cells of the early embryo.
Quiet.
Yeah.
Rolling. Hold for speed. And speed.
This afternoon of Friday, February 1, 1991, we're in the second floor library of the Beckman Center at Stanford University, talking with Doctors Leonard and Leonore Herzenberg, Len and Lee. Very interesting couple and unique in quite a number of ways of having worked together from their very early careers. And maybe we can start with that. Leonard, tell us how you met Lee and how your careers started.
I met Lee while I was at Brooklyn College, where I was a senior, and I guess you were...
Entering freshman.
Were you entering freshman?
Yep.
Yeah. And then by the end of that year, we'd fallen in love and decided we'd get married, but not immediately. I'd go off to Caltech for graduate school, and Lee would stay at Brooklyn until she finished her degree, was the plan. We actually had a summer together, though, because I had a fellowship to go to Woods Hole Marine Biological Laboratory on Cape Cod, take the invertebrate zoology course. And Lee came along. The only job she could get, I think, was some kind of
I was a chambermaid. I was
Cha-
an exalted chambermaid.
she made beds.
I made beds.
But we didn't sleep that often in the in the beds that she made. The beaches were very nice. That's how we met. Another
But
question?
then you went off to Caltech by yourself, right?
Yeah, I went off to Caltech by myself, and I spent the first year there and came back the following summer when we got married. During the year, it became obvious that we'd be better off married than not married, staying apart. In that way, we'd be much happier. So Lee came out to, well, I went back to Brooklyn and we married there. I worked for the summer at a hospital laboratory. It was actually kind of interesting because they'd gotten lots of money. This was in 1953, post-Sputnik. And they had all this equipment that this hospital laboratory had bought, and they didn't know how to use it, by and large. Now, I didn't either, but I at least had 1 year of graduate school under my belt. And I had great fun that summer putting all the equipment together, running electrophoresis, and they did know how to run the optocentrifuge. That was the one thing they had done before. But I ran whatever equipment that they had bought, I put together and tried it out on something. Then we drove back across the country to Caltech to continue graduate school, as I recall it. I had to continue graduate school. And is Lee going to tell part of the story?
Yeah, sure. Well, I was going to ask you, Lee, what you found when you went out there, and since you had to cut off your schooling at Brooklyn College. What was your plan?
I was already accepted at Pomona College. So I went to Pomona College for the first semester, and that was like a drive. At that time, there were a lot of orchards and stuff, and the place was foggy. I'd get up at 6 in the morning to drive, and I'd spend an hour cutting through that very heavy fog that they used to have. But mostly I found that what was being taught at Pomona wasn't very exciting, because I'd come home from school and I'd go straight to Len's lab, and I'd go to seminars at Caltech and learn what was going on there. And the distance between what was being taught and talked about at a really first-rate institution was so far from the biology classes that I was taking that eventually I became this very uppity undergraduate and said, well, I don't want to learn this. I mean, this is very silly. This is just teaching me to be a technician. And I can go to Caltech instead and sit in on the seminars.
Why couldn't you go to Caltech as a graduate or as an undergraduate?
Good question, because they didn't admit women. Actually, none of the major institutions admitted women at that time. It wasn't any great shock that they didn't admit women. It was an annoyance, but it was something that you just accepted. You know, well, they didn't, and maybe they would sometime.
Well,
but
can I interrupt at all?
Sure.
I mean, many major institutions did. I mean, the state universities, large universities all over did admit men and women, but there weren't.
Harvard, Yale, all the Ivy Leagues didn't admit women, but they had sister schools. So Caltech didn't have anything like that, and the closest I could find was Pomona, which was quite a distance. That was hard, tough stuff. I was 19 years old. I'd never been out of New York. We were ghetto kids. I mean, the whole view of the world was completely changed. The only time I'd ever been out was when we were up in Woods Hole, and I learned a little bit about what I was going into from that, but it's a very different world. But the professors at Caltech, as opposed to the Caltech administration, were extremely hospitable. And they let me take all the courses, so I took whatever courses there were. Essentially, I moved from undergraduate into a graduate environment because there was no real undergraduate environment in biology at Caltech at that time. And I had a wonderful time. I mean, I took the greatest classes. I had the greatest professors. I mean, the list of people that were at the department at that time ranged from Renato Del Becco, Max Delbruck, George Beadle, Sterling Emerson, Ray Owen, Albert Tyler, for whom I worked, Henry Borsa. So it's a wonderful...
Alfred Sturtevant.
Right, the people on the third floor, right, and Ed Lewis. And Norman Horowitz. The list reads like a who's who in science. I mean, these are all names that anybody who's been in science, when I call that list off, they just go, wow, wow, wow.
But besides taking the courses, you also were helping Len with some of the work that he was doing at that time, too, or towards what getting the dissertation.
Well, slowly Lee would be hanging around the lab, but she also was working to make some money. We had very little money at the time, and studying. But I think as time went on, Lee would act as a technician, keeping some records. But when it came time to write my thesis, Lee helped a lot, both in the typing of it and in, I can't remember how much, but probably in a fair amount of the fixing up of the writing of it.
I can't remember that. I don't know
It may not be true at that point, but later on.
I know I drew all the figures, because that was one of the ways I was making money, was drawing figures by everything that a Macintosh can do in 15 minutes, I did in weeks. I used to take people's theses in and then just run the whole thing and do all the drawings. I don't think I actually did any lab work per se, except just occasional watching an experiment if something had to be watched. With Len until well after we went to Paris, and mainly he was a very good technician. He really did not want my hands involved in stuff. He was not certain that I was going to handle the things the way he wanted them handled.
Also, I was doing chemistry by and large, or kinds of things that didn't require a lot of replication, but it required one after another, one step after another. Not the kind of thing you normally need a technician for.
Quite frankly, you really did not trust me to do that work.
That could be.
And I didn't disagree. I mean,
Stand by for speed. Speed.
After you've completed your graduate work at Caltech, you did your postgraduate work, at least 2 years of it, at the Pasteur Institute, and you both went over at the time. And if I remember correctly, Lee, you were even pregnant on that boat trip over. Could you talk a little bit about your stay in Paris and your collaboration there?
Sure. Yeah. Well, we did. We had the opportunity to go to France by the old Liberté. No, the old Ile de France first, so by boat, from New York. And that's something which nowadays there's no opportunity to do at all. We almost always fly. I haven't taken a boat since returning from France later on in the Liberté.
mind you, we were 5 stories below the ocean, in the littlest cabin you could ever imagine.
Yeah, and Lee was feeling nauseated. We thought it was the being on a boat and being well down in the tourist section and so on. But went to a doctor, I remember, and the doctor said, "Oh, you have mal de foie." That means liver sickness of some sort, which is what French doctors always said you had if you had a stomach ache or a headache or something. They would call it mal de foie, anything. But what happened when we arrived in Paris and you went to another doctor? He found you were pregnant.
Yeah.
Yeah. So it was the early month of pregnancy where you do feel nauseated normally, so it wasn't because of the boat trip per se. Anyway, we arrived in Paris by taking the boat train from where the boat landed, I think in Cherbourg, wasn't it? Or anyway, Le Havre, that's right, Le Havre, and to Paris by train. I think we were met in Le Havre by a childhood friend of Lee's, a woman who had lived. Well, Lee can talk about this more easily than I can. We're taken to it was very hard to find a place to live. There were no apartments. So we were put into a hotel, in one room, and it was cold there. And one of the wedding presents we got was a heating blanket. And so we put the heating blanket on and found that the matron from the hotel would come upstairs or would telephone us and say, "Why do you have the lights on so late?" And we said, "We don't have the lights on, we have this heating blanket on." Heating blanket, you're not allowed to have such thing, no heaters in the room. And so they cut off our electricity. They do it room by room. The electricity was controlled. There was a little light that showed at the desk that the electricity was being used in the room. I had a cold or something. In fact, I was feeling miserable.
In fact, you had pneumonia.
I had pneumonia. I had pneumonia a number of times in my life, and found out why, I think. And it was pretty miserable there in that crummy hotel room, which is the best, well, the only one we could find and afford. We lived there for about a month or 6 weeks.
No, more like 4 months.
Is it that long?
Yeah, I was fully pregnant. We finally decided we had to move somewhere because we were going to have a baby. So we moved outside of Paris to something they called an espèce de chateau. It was a wonderful children's apartment in an old chateau house that was run by some crazy ex-dancer who'd been the mistress of some Italian, very wealthy Italian industrialist. And he bought her this chateau, but she didn't have any support money. So she lived in one corner of it, and she let us rent the other corner. That was quite nice. Except it was a distance outside Paris, which meant that it was hard for me to get to the lab. I either had to go in the morning with Len or not go at all. I stayed home a fair amount of time at that time. There have been a few periods in my life when I didn't work, but very, very short periods, namely. When we first got to Paris, Len said, well, now you can do all the artwork that you want and go to the museums and do all that. And after about a week of that, I came crying, saying, please let me come to the lab. I'm pregnant and I'm lonely and I don't want to do all that. We worked with Jacques Monod then. He was of the Monod and Pardee and Jacob Leboeuf. Jacob Leboeuf, yeah. Anyway, that whole group, which later became the basic foundation for molecular biology as we know it today. And we were there just at the dawn of that. In fact, some of the experiments Len did were the experiments upon which the whole system rested, that they were then able to go and do all molecular biology in.
Yeah, I talked about that quite a bit previously on these tapes. I guess we're supposed to be talking about sort of our.
Yeah, but then you also, Lee, you mentioned that part of the ritual was this long lunch where a lot of the ideas were discussed, and that you were part of.
Well, from my point of view, I mean, for reasons that, in retrospect, really amaze me, but people were very, very good about letting me just come to lab seminars, join in the lunch. I was just taken as one of the gang and allowed to ask questions or talk or pipe up with my opinions or whatever as things went on. We had lunch together every day at Pasteur. There was like a small solarium. In fact, it had been used in the old Pasteur times as a place where they kept the wine vats and did all their fermentations, and it was heated by the sun. So it was a very nice little lunchroom that we had. And one table of that lunchroom, there were two tables. One would be the technicians who would always come in and cook their dinner, their lunch. They'd heat them up in little pans and do a typical French thing of having a hot lunch with the regular food they'd bring in from home. And then the scientific staff always sat at the other table, and we would go out to the charcuterie and buy milk and ham and something, make a sandwich out of it. A few people would bring things from home, but usually that group ate that way.
French bread was the most important part of it.
Yeah, God, you couldn't live without French bread. But it was a very interesting time. I think I mentioned to you when we were talking before that politically it was interesting. These were people who had been members of the resistance. They had close links to Jean-Paul Sartre and Camus, the existential movement. It was a small intellectual community in France, particularly on the left. And they were all leftists. They would tell us stories about how they hid various Jews who were part of the group at different times when the Gestapo would come looking for them. And, it's a very strong sense of not communism. I mean, they quit the party. Most of them had quit at the time of the Hitler-Stalin Pact, some of them sometime earlier. But they were socially oriented. I mean, they were very, in these days we'd probably call them liberal democratic. I mean, they were very concerned with human rights in all ways, and they were concerned with scientific rights, the rights of scientists, communication among scientists. And that was something that we had done. We'd already been into the Federation of American Scientists. We later came back, when we came back from Paris, we ran the Washington office for a couple of years while we were at the NIH.
Well, I was the treasurer of the national organization, but the office was to say we ran the office when
Well,
many distinguished scientists, particularly atomic scientists, people who worked on developing the atomic bomb.
They ran the organization. We ran the office. That's what I meant.
Yeah. Well, we put out a newsletter.
Yeah.
And,
We did the mailings. We did, the things that young people do in political organizations.
When you came back to Washington, you were both at NIH, but there you sort of split, right?
Yes.
And you were at one lab and...
Yeah. Well, maybe we should say one more thing about Paris. I mean, I had a salary, and Lee didn't. And there was no formal. She couldn't get paid at the Pasteur Institute, but we managed to live fairly well on my income. It was only when we had to come back to the NIH that we should have had our ways paid and all of our goods brought back, but I was actually inducted into I was examined and then inducted into the public health service in Paris, and
That's in lieu of going to the army, right?
That's right.
Choice,
Yes, he carried a pipette for his country, as we used to say.
That's right. But the question I'm dealing with, which may be of interest to future historians, is that American scientists lived quite well in Paris in the early mid-50s as compared to French scientists. But when we had to go back to America, it was a financial loss. We had to borrow money to be able to get back because the public health service didn't pay for our way back. I think mainly because Harry Eagle, to whose lab we were going, didn't want to pay for it out of his laboratory budget. And so we came there with no money, with considerable debts.
But I think I'd quibble with you about living quite well. I mean, we didn't live we went once to a fancy French restaurant in our whole time, and had the whole time spoiled by the ugly Americans who came into the restaurant at the same time. And we'll never forget their comment was, "Did you see the Mona Lisa?" "Yeah, I was really disappointed. It's so small." It was. You just remember this because we really didn't have money to do anything when we were there. I mean, we ate, but we had a nice apartment that we were lucky. The Pasteur found us an apartment that belonged to someone else, and it was expensive enough, but not that expensive. But I wouldn't say that. I mean, living quite well is another. Well, this is a dynamic between us, okay? I mean, all of our lives, we've always shaped and honed the idea between the two of us to get exactly what we mean out of it. We're also very intensively measurement-oriented, and I remember explicitly driving through Kansas, and we were trying to estimate the number of feet between the lampposts on a long, thin road that did nothing but have lampposts on it. Telephone poles, wasn't lampposts. And we were trying to estimate that, and our conversations have always been honing things to as fine an understanding between us as we can come. There are arguments, that's another issue, but these are more like clear discussions, but I don't agree with you that we lived quite well. I think we kind of lived well.
So speaking of the dynamic, it sort of brings to mind, and maybe we'll hopscotch a little bit here, but maybe back to Caltech and I guess to your meetings with Barbara McClintock, and what I'd like you to say a little bit more, what you told me yesterday, Lee, which I found very interesting, is how Barbara McClintock looked at the world and organized her facts and theories, and your explanation with the baby and the milk, and how boys and girls are trained differently, because I think that's quite a good illustration and analogy of different ways of looking at the world.
Well, the story that you're referring to started one Friday night when Len and I were just about to go out to a Chinese restaurant at Caltech. And that meant driving in and finding a cheap student restaurant in Chinatown in Los Angeles. And since we were going to take that whole drive, we were kind of wandering around the place looking to see who wanted to go, and there was no one there except for Barbara McClintock, who was sitting in a quiet room with the light on, and she was obviously working. It must have been about 8 o'clock when we were looking. And we stood outside the door and we said, well, you know, should we ask her or should we not ask her? She was high-level faculty to us, although she was always this very simple and very direct woman, but nonetheless she was part of the faculty, and we were just students, and we hassled a long time back and forth saying, well, we shouldn't interrupt her. She's obviously busy. Well, but maybe she really would like to go. Maybe nobody ever asks her. And this kind of thing. We've learned that ourselves. I mean, you know, we see the students will sometimes go out and, you know, we wish they'd say, come along with us. But in any event, we finally did get up the courage and ask her, and she said, oh, I'd love to go. And what I didn't tell you when we were talking about it last time was that many years later, I would always, every so often, go and visit her. And I'd start out, Dr. McClintock, you probably don't remember me. And she said, what's your name? What's your name? It's Lee Herzenberg. Oh, yes, we went to that Chinese restaurant, didn't we? It's really fun. She's a lovely lady. But the particular question was, how does Barbara McClintock see the world? And I asked her at the time, she was doing what was the Jumping Jeans story. And the rumor at Caltech was that she was the only one who really understood this, and a couple of other professors could follow what she was saying. But for the most part, people couldn't really follow exactly. It was just such fantastic work. And my own little way, you know, how do you find these novel things that nobody else has found? And her description was that as she's doing things, she tests the observations that she makes against the existing paradigm, the theory that's currently being worked. Most of the observations fit into the theory, and therefore they just extend. It's what she expected to happen based on the theory that she was working with. But every so often, she'd run into an observation that she was certain was true. You know, a blue kernel in a otherwise yellow field, which shouldn't have been there. And there was no explanation for this. But you can either shrug your shoulders and say, well, you know, these things just happen. Or you can say, I know this happened, there has to be a mechanism. And so she would look at it and decide that it's technically correct. I'll store the information. And she stores that observation as something then that she can't explain with current theory. And then as she gets another one and another one, she tests against what she's got stored until she begins to group observations. And finally, out of that comes a method of explaining how this must be happening. Actually, what happens is you don't do this as consciously but that's, if you look at the process, that's what you're doing. Eventually, it gets niggling, it gets annoying enough to say, this can't be correct. It can't be the theory. There's something wrong with the theory, because I know that under these conditions this happens, despite the fact that the theory says it can't happen. And so I've got to find some explanation, and you go to sleep at night, and you think, and you think, and you wake up in the morning, and you think, and you say, well, if that's true, I could do such and such an experiment, which wouldn't be predicted by current theory, but would be predicted if I include these new observations and extend the theory. And so you're saying that's how she basically functioned. And in later years, I really realized that she had shaped my thinking. I thought that was great. That was a perfectly logical, rational way to go about the world. And I still think most good scientists think that way, male or female. But it's been put by other people that was a particularly female mechanism for dealing with the world. And so then came the question of, you know, what is the difference between the way men and women look at the world in an intellectual sense? And that was the point you were raising about the milk, that by my observations of, you know, growing up, and I grew up as a ghetto kid in a Jewish family, but I don't think it's terribly different anywhere you look, that if you had a little boy sitting at the table and he was sitting there doing his homework and his mother said, I just put a glass of milk to warm for you on the gas stove, and keep an eye on it because it's going to boil over, I have to go out to the store, or I have to go in the other room. A little boy would sit there doing his homework, and he'd forget about it, and the milk would boil over, and Mama would come home and find that the stove was full of milk. The kid would be drinking the milk, but the stove was full of milk, and he hadn't even cleaned it up. He hadn't done anything with it. He'd gone back, and he was sitting there and studying his books. And she'd come in, and she'd say, you know, you're supposed to, you know, why did you let the milk go there? But You concentrate so heavily, you're so intensive on these books, you never can think of anything. And she'll tell everybody the story about how little Joey is concentrating so heavily, he's so intense, he's so interested in what he's doing, that he can't even recognize that something like the milk is boiling over on the stove. Alternatively, if his sister is sitting there and doing the same thing, concentrating just as heavily, and Mama comes home, the sister gets a good smack and says, you're supposed to know that. And she says, but I was doing my homework and I don't care if you were doing your homework. You got it, there's milk on the stove. You're going to leave it that way to boil over. You can't do that. And that extends it to training that's almost necessary if you're thinking in terms of the girls growing up to rear children and the boys growing up to work. Because when you're in a house with children, if you were concentrating heavily on anything else, the children could take poison, they can fall out, fall in the well, whatever. You've got to be able to keep your eye on peripheral items at the same time as you're concentrating on what you're concentrating on. And Len sometimes remonstrates with me that when I read a book, I get so intense in the book that I can't hear him when he talks to me. He doesn't realize, of course, it's true for most everybody.
Len, do you have any thoughts or arguments with Lee about this? Do you see any gender differences in the way people view the world or do science and look at the organisms? Because there's a sense that
so far as I think the only lesson from what Lee is telling about the milk story is that women are, she's saying there's a gender difference in that women are not allowed to concentrate as intensively as men boys from early childhood, because the girls are supposed to pay attention to things around. I don't think that's true in general. I think it's true about Lee.
I think it's not precisely what I was saying, though. What I was saying is that women are forced to include peripheral vision. It doesn't mean that you can't concentrate as much, but you have to keep your peripheral vision functioning. You're not allowed to shut out the other items. Does that make sense?
Well, one of the things that characterizes our lives is that when I try to repeat what Leah said she said, she always disagrees with it. So I think that Leah's a good scientist. I think to try to analyze our interactions would take, a life study, probably on a psychiatrist, psychologist.
Maybe we could talk a bit of how you plan your research projects and how is it determined what Leonard would be working on, what Lee works on. Tell us a little bit about that dynamic.
Well, I guess I would characterize our scientific lives not so different from our non-scientific or outside the laboratory lives. That to a large extent, what I am excited about, what I want to talk about is what I'm excited about. And Leah's the person I want to talk to about it because she's a very bright person and fun to talk to about it. And I always get feedback. Sometimes one could call it arguments. And certainly Lee started in my laboratory. I was the one who had a PhD. Lee had no degree. She didn't even have a bachelor's degree. But she was intelligent, and she wanted to understand what it was I was doing, and I wanted to tell her about it. It was more fun telling her than others in many cases. So we really started working together seriously in Paris, where Lee would take the notes on experiments that I was doing, or she would read this spectrophotometer to read the values, which would tell us how much beta-galactosidase was being made by a culture. And we get lots of data collected. Lee would keep the notebook, and I would do most of the pipetting. I think I did most of it.
You did all the pipetting?
I did all the pipetting at that time. But there was very funny business at the Pasteur Institute. The experiments required the use of large test tubes, and with pipettes sitting in them and then being in a shaking water bath where they went back and forth. So you kept the pipettes in the test tubes. And those test tubes were not uniform. And some of them would, after a while, have the pipette go through the bottom of the test tube and you lose all the contents. That would destroy the usefulness of that tube. I think one of Lee's first jobs was to help, and then maybe she did more than help, she maybe did a lot of it, was to bounce the test tubes on the marble table, which is from a certain height. And if they didn't break, then we could use that one. If it broke, we threw it away. That would indicate how, that we had enough strength at the bottom of the tube. Remember that?
Oh, I remember very well.
I think you must have spent many hours, many hours,
It was my brilliant invention. I could figure it was one of our usual arguments. I can do this, and if I do this, you won't have the tubes break. Don't be silly. Yes, it's going to work. You'll see.
Well, I don't remember that detail, but so Lee kept many of the records, and which was fun for me because I don't keep records really well. I don't like to. I don't like detail work. I don't even like writing too much.
You don't like to write anything on paper.
And Lee loved to, to do that and to worry over it and to improve it and so on. And I like to, I don't know, come up with ideas which intrigue me, which I think are good ideas, and to follow through on that. But the detail work is not, particularly the writing and the record keeping, is not really my forte. So I think that when we went on that way, we spent a lot of time thinking about what the consequences of the results were. And Lee would worry more about many of the details than I would. I'd say that I'm really not interested in details. I'm interested in the bigger picture. I don't know if I said that in words, but that was really the feeling. And
That's your Barbara McClintock issue there, of the things that didn't fit exactly. I wanted to know how they fit, or I was storing them away and bringing up, but this can't be with this. And it was a use. I mean, I had a good memory for all of these crazy items that had not fit into the particular framework that we were functioning in. And I don't know if it's, I think you sell yourself short in saying that, I mean, you're an excellent detail person, an excellent, I mean.
Well, I'm a very good editor, I think, which means details. I do catch errors pretty well, that other people do, but.
And I don't.
Lee's not as good at picking up errors frequently, I would say.
I can't proofread worth anything. I mean,
But also, maybe we come to the current day because it's hard to really remember back there, but I think the roots of it, I don't think things have changed very much in many respects in our styles, that I'm always looking for something new to do and to sort of make a sort of major contribution, you might say. And I don't like to fill in all the details. I don't like to dot the I's and cross the T's too much. Although I like a firm result. I mean, I guess if I had started working now, I'd say that isolating a protein or isolating a DNA, a gene, determining its sequence would give me a lot of pleasure, and it has in the past. I mean, just to actually get the exact structure clearly. Theories are, for me, not as interesting unless they're very tight, at least I think that way, unless they're tightly defined theories. In other words, I don't like what I consider idle speculation. I don't think I'm not creative, though. I think I am creative because there are many different things that I've done that have turned out to be very useful and informative. But I've also worked in many different areas because I like to work in many different areas. Lee, I would characterize as much more of a completer. In fact, we used to read a book called the I Ching, the Book of Changes, Chinese fortune-telling book, sort of Chinese naturalist book. And it's a little bit long to talk about exactly what it is, but there were two characters in it. There was the creative and the receptive. There was the heaven and earth. So heaven would be the creative, and earth would be the characteristic of the receptive. But each of us is made up of part of each of these two opposite characters. But Lee I'd characterize as being more Kuhn, at least we did for a long while, which is the receptive, and me is the more creative Chen, I think it was called, wasn't it?
Yeah, well, you have to
Chen. But at times
temper that a lot because it isn't. It depends on what your definition of creative is.
That's true.
I mean, I have always characterized you as someone who sees the development of a new method a new way of doing something as the potential for opening your way to getting answers that couldn't be gotten within the limitations of the technique. That came to the facts. It was the idea of generating the facts. It was the concept of using monoclonal antibodies as reagents for the facts, which was, I think, absolutely and clearly Len's contribution to that. And hence, all the stuff that follows from that, including the CD4 analyses that we see now, is all based on those very direct concepts. Seeing the method was not adequate. He's not a methods builder per se, because methods builders don't deal with the science. It's seeing where the science is bounded by its methodology and saying, okay, we've got to somehow strike to break that particular barrier, and if we can break that barrier, then we can get answers that are very special. And that's what I see you as forging ahead and doing all the time. And every time that he's gotten the answers, these have been first-rate science. You don't publish them in methods journals. You publish them in Nature and Science and PNAS. And, they're really first-rate introductions of things to science, but they're only possible because you've broken the methods barrier. Once he breaks a methods barrier, I will see an infinite number of places where in the areas that I'm interested in, I can use that method to do what it is I really want to do. And so in that sense, I'm the completer. But for many, many years, I think as long as we've been at Stanford, I've worked on a particular area and a particular set of projects. And there, the big fight between us is that it's a much mushier area than Len would choose to use. And I tend to like to create theory. And the latest of those is a concept of evolutionary layering of the immune system, that rather than having a single stem cell that evolved and gives us the entire immune system, what we've done is retain through evolution a series of progenitor cells for the immune system. And as the fetus develops, you first kick in stem cell number 1, and that gives you the basic fetal hemoglobin and fetal kinds of lymphocytes, and then you kick in stem cell 2, and it gives you an intermediate. And you finally work your way all the way up to the top stem cell, and it kicks in and gives you what we as mammals use as our, not primary, but high quality defense against invading organisms, high affinity, high quality. Anyway, developing this theory is this is the kind of theory that I like to work on. Or
can I interrupt and
yeah, sure.
comment on that? It's interesting that you picked that particular theory as being one which I wouldn't.
Well, you do like that one, actually.
That is the theory which I think perhaps you had more to do with the creation of, but that maybe much more to do with. I'm not really sure. But we published that paper as a review with just the two of us, no other people, which is unusual if you look at our publication record. There usually are other people involved in any of our papers. If we're both on it, there's always other people. But this one we just wrote the two of us. And I enjoyed that one, and I actually tell people about that theory when you're not around.
Yeah, I was going to say that there's a theory that I put together with one of my students that has to do with likening certain kinds of immunoregulatory processes to flip-flop circuits in the electronic circuits, which I think essentially gave us the guideposts to discover and work out a whole system of immunology that we couldn't have ever understood. It would have been in the McClintock range of ununderstandable observations, except for the fact that we happened to have put this theory together before with only the vaguest sense that it might be really relevant. And that was an issue of great argument between Len and me, and most of our colleagues, actually, in the European Journal of Immunology. We submitted it there, and there was apparently a grand battle at the editorial level, with most people saying this theory is just too vague, and we don't want to publish it. And Niels Jerne coming in, apparently, and saying that he thought it should be published because it made sense to him. And so they published it in volume 10, number 1, page 1, which was 10 years to the date of the location of Niels Jerne's article that is far greater in theory. I mean, he really had some of the key
On
theory on the
idiotype
idiotype and
idiotype networks, on networks.
yeah, the word I was looking for was complementary idiotype interactions.
But it was on networks.
Right. And this was an extension of network theory, which is why the European Journal was the right place to put it.
May I interrupt now? Or do you want
No,
to?
let me just finish 1 second. That the joke about that was basically that either somebody thought it was a good theory, or else once every 10 years in that slot they can publish something that they think may be junk. I don't know the answer. Well, we never came to agreement on that theory. You still hate
No,
that theory.
that's a very good example, I think, of a non-theory, which, and Niels Jörner's idea is also one that's very, very controversial as to whether it really is a theory or not. Most Americans, it's interesting, most Americans basically don't follow the network theory of the immune response. Don't think of it, don't design experiments based upon it, and whenever it's talked about, tend to call it with derogatory words. I'm being very careful about my language for history.
I think one thing that...
But I wanted to, if I
sure,
could finish for a
sure.
second, which is that the Europeans, by and large, like the theory, like the idea, and I don't think that the idea is very, very useful either. Either the network idea per se, although there's a lot of kind of things which you can get to follow it. I think the same is true of what Lee calls the bimodal regulation. I think bimodal regulation is obvious that that's true or not true.
I call it bistable regulation.
Bistable to the bimodal, bistable regulation. I think it's a trivial idea, and it doesn't lead to experiments. You can describe many situations as being due to bistable regulation, and if you don't describe it that way, you can figure out a way to do an experiment so you can describe it that way. That's basically my feeling about it.
Yeah,
So we've agreed to disagree on this for a long time, and
No,
I think that's true to now.
The answer to the network theory is that it's been completely predictive of the work with Lylan B cells, although not with other B cells, which again is work that I've been doing with my colleagues. But secondly, that without the bistable theory, you can't really explain epitope-specific regulation. And as a result, epitope-specific regulation, which is like we have 6 or 8 papers on this, it's been reproduced, it's very important to vaccine makers, but amongst particularly American immunologists, they refuse to accept the data. They just forget it every time, and they forget it because there's no current theory that will explain it except this kind of bistable theory. And so the easier thing is to just say, well, the data doesn't exist. And that's actually a fascinating issue in terms of history of science as to how experiments get done that don't fit the paradigm and simply get lost because people can't explain them, and they just shrug their shoulders and say, well, there was some artifact involved in that. And for us working through that, the work, it was very easy because the theory was there. It provided all the guideposts we needed, and we could predict everything that was happening based on this theory. And you knew this at the time. You've forgotten it since then, or I don't think you would make the arguments that you make. But we can go back over it sometime. Maybe you would anyway.
Well, I think what I wanted to say that it's been quite clear from our brief conversations here that your work goes on and you're clearly thinking and creating, although somewhat we've been talking in reminiscences in history, but I think today's New York Times article has been sort of fortuitous for us because here it announces some of the new work that you've been doing on AIDS and presenting further avenues not only of research but of clinical application. And maybe we could end with a few words on that. Your reactions to the article, your work dealing with AIDS.
Well, the article, should I start?
No. Let me start, because I want to say something.
Who's going to
Very
finish?
exclusive. You may finish.
Who's going to finish?
You may finish. No, the reason that I don't think you will do yourself justice, and that's why I've asked to start, in that this is a perfect example of a place where Len had set up a method because he could see that understanding the regulation of gene expression was being hampered by methods where you couldn't look in individual cells at whether a gene was turned on or not. You had to take a culture, mash the cells up, get the extract, and then say, well, okay, the gene was turned on to some extent. The implication being it was turned on to some extent in all cells in the culture, but it could have been turned on for only a few cells in the culture to a very high extent, and you wouldn't know the difference. So Len set up...
Excuse me, this is described elsewhere in the tapes. So
Yeah, but this is
Yeah,
a
but
good
this is-
we'll go on then.
Yeah.
So Len set up that method and was making good progress with it in a number of areas when, and had already set up the HIV, the human immunodeficiency virus, the AIDS virus, taken the regulatory sequences from that and put them in front of the sequences for the enzyme that would allow us to see what was going on if the gene was turned, if something would turn the virus production on, it would turn on the production of this enzyme. So it gave us a rapid, very accurate cell-by-cell assay of what we're doing. And I mean, that's where, his knowledge and his understanding that was going to be a very important method for doing all sorts of things was there, and he was now making progress in one area when we heard from Tony Fauci that there were human cytokines, the things that are produced during inflammation, that were turning on the virus. And Len said, well, it's clear, we said together, we were both in Berlin, that it's clear we can use we can use the assay that Len had developed to get a much cleaner picture of what actually was going on when these cytokines were stimulating cells. Applied it to that, and then took a drug that I had been sort of playing with in terms of, that's a bad term, playing, but trying to figure out whether this might be good for AIDS patients. There were some theoretical reasons why it might be, along the kinds of theory we were talking about before, and I thought they were valid, and I really wanted to go with them. Len was questioning just how much use there was to this, but on the other hand, he was certainly interested in it. But when he picked up and could see his way clear to doing the set of experiments, the hard, tight experiments that he likes to do, then he took this over and he just picked up the project and really ran with it and created what is there. And I came back into it because I didn't know very much molecular biology when we started that project. I did all my learning as I was following him along, just trying to help out with the students and trying to help out with the clinical data. Okay?
You got the last word.
Stop there and change.
Whoa, that's too bad. Len, we gave you the last word.
I'm sorry.
Wahoo?
Yeah.
Titan.
Okay. Good. Okay. Thanks.
All right, go ahead.
Len, we gave you the last word on this, so go ahead.
Well, it's not that I really need the last word, but I'd like to, in this particular case, I'd like the last word in other situations, is that this is a very interesting problem. This article in the New York Times is reporting a paper which will appear shortly in the PNAS, a paper that involves a collaboration between Anthony Fauci, Tony Fauci, and what's his name?
Meister.
Alton Meister. Don't let him hear that part. I should know him. He's very well known. He's Mr. Glutathione, and he's worked on glutathione for many years and its involvement in various aspects of metabolism. Glutathione is a very important molecule. It's a tripeptide. It's got an SH group on it, and it's a major antioxidant in cells. It's present in cells, virtually all cells, at millimolar concentrations, which is a very high concentrations for small molecules. By comparison, the usual nutrients, the usual amino acids are found at a tenth or a hundredth of that concentration, or even a thousandth of that concentration. When we got involved with, after we heard from a German scientist named Wolf Droger, that glutathione levels are low in AIDS patients. Data wasn't very good, but it was good enough for us to to think about it some more because we were actually very concerned about the lover of someone we knew very well who was dying of AIDS. And we had AIDS on our mind a lot. And here came something which promised some possible useful treatment. Not that it would save his life, but it might extend his life usefully or help others at some point. The reason for this logic was that glutathione levels were low in AIDS, and there was a simple drug that had been used for many years that could raise glutathione levels back to normal. And since glutathione is such an important molecule, as has been shown by the years of work by Meister and many others, that if you're lacking adequate concentrations of a very important molecule, and you can restore it without any danger to the patient or the person, you should do that. And that was something that Lee felt very strongly, I believe, before I did, but nevertheless, I mean, it wasn't by more than maybe microseconds or maybe days. But,
Well, it was very much a we operation.
Yeah,
I'm just saying that it was something I was carrying the ball more for, if you like. In that
well, Lee was okay. So Lee was doing. We decided to. This drug, by the way, was available in this country, but not in an orally palatable form. That is, it had to be injected IV, or it tasted absolutely terrible. This drug, N-acetylcysteine, and so we couldn't get somebody to take it more than once before he basically was his stomach would be very upset. And it tasted so terrible. It tastes like rotten eggs. And but in Europe, you could get it as little capsules or tablets rather, which if you broke them, put them in half a glass of water, they were like Alka-Seltzer, carbonated beverage. Tasted okay. And it came in another form too, where you could taste it like Dilute Tang, which is an orange-flavored, non-carbonated drink. They tasted all right, so people could take them 3 times a day without any real discomfort or nausea or anything. We found out why. So we imported the NAC ourselves from this German scientist who had told us about it and the lowered glutathione levels, and he sent it over to us, and we gave it to our friend, and other people asked for it, and other people who had AIDS. Lee promoted it to legally, but not, but gave it to them.
But we talked to their physicians, basically.
We always talk to
It was always
physicians
done with
first.
the physicians,
That's right.
because it doesn't make any sense otherwise.
Well, we thought we saw miraculous improvements in some people who had AIDS or who had pre-AIDS or who had, and we made arrangements with a company. Don't have to go into all the details, but that they would, the company, a European company that made the drug, and they made it cheaply, and we entered into a contract with them based upon a patent that we applied for. They wouldn't raise the price of it if it became useful for AIDS. So then we went in 2 directions. Lee continued to talk to people with AIDS or who heard about it. It got written up in the AIDS newsletter and various literature. The grapevine spread the news. We had a press conference because I happened to get invited to I had been invited a year earlier to a high-class meeting on AIDS. And I was invited only because I was a well-known immunologist. And actually, to tell the story a bit further, I was invited by Lewis Thomas at the advice of Jerry Lawrence, both of whom I'm going to see Monday, where I'm going to give a major lecture at NYU, where these people are. And you can look up Lewis Thomas and Jerry Lawrence if you want historians of the future. But I talked about results we'd gotten, which I had started working on in the lab, as Lee partly described, that we found that HIV, the virus, either in the construct form with galactosides as a reporter gene, or the virus itself later on, would be inhibited by this N-acetylcysteine, this drug, NAC, which restored the glutathione levels. That wasn't an expected result, but it was a clear result, and we confirmed it in many ways, and we published it. But before we published it, we went to a meeting where Tony Fauci was giving a lecture, and we talked to him. We spent a lot of time with him, and he told us about these cytokines stimulating the virus production. In particular, TNF, tumor necrosis factor was called, or cachectin, whatever it is. The three initials describe it perfectly well. And the drug we were working with, is N-acetylcysteine, would block the action of TNF in stimulating the virus. This is real interesting molecular biological results, real good gene regulation stuff, but working with something which was of crucial importance to the world, because there's a real scourge of the world to have so many people with AIDS and so many more expected to get it. And there is no cure on the horizon. So it was very exciting intellectually to do these experiments and get such clear-cut results, and we did many more experiments. At the same time, it was very exciting to have the feeling that if a clinical trial got done, which was the only thing which would really establish whether it was useful in treatment, there was a good chance it would work, we felt. And we felt, as we went on, we felt more and more that the chances were better, because we found more and more people taking it and doing pretty well with it. At this point, we feel very strongly that there's just got to be a clinical trial. This is now 2 years after it was started. Well, Tony Fauci, who is the head of the AIDS effort in the United States at this point, at the NIH, is also quite committed to this clinical trial, and he's going to be carrying out the first one for reasons we just have not succeeded in getting the medical or the drug community or the FDA to further the idea of the clinical trial. Even though there is a company that would make a lot of money out of it, they think that for a variety of reasons they've held back. And so it's 2 years, and it still hasn't happened, more than 2 years. Now, I'm in touch with that company a lot, and I'm in touch with other people, and serious people have laughed at the idea that such a simple compound, glutathione, or that N-acetylcysteine, which restores glutathione, could be useful in treating this disease, which the best minds are finding only poisons to treat this disease with. So we're rather emotionally involved with it as well. But I think I really do think I'm maintaining my scientific objectivity in the laboratory work, and the clinical work we're just trying to get done by these trials being done by others who are doctors who can carry out clinical trials effectively. It's their business. It's a very interesting example of where there are people, there's a lot of money in AIDS, therefore a lot of people have gone into working in that area, and they're not always the best people, but even if they are the best people, they're so imbued with working on very technological solutions, on developing at great expense crystallographic structures of a piece of the virus, and they might learn something from that which will allow them to get another drug, or they've bioengineered DNA, recombinant DNA manipulation, and so on. Best, latest techniques, and they get them into clinical trials, and they find they're of no use. They spend millions and tens and, a lot of money, up into the billions, actually. And there are billions of dollars a year in profits we made on any of these things working, too. So it's an interesting place to be at the age of 60, being a top scientist. I've been very well recognized as a good scientist. Leah is well recognized, even though she doesn't have written recognition. She only became a PhD fairly recently, as you remember from the tapes. And she became a professor fairly recently, so she's been in my shadow in some ways in terms of recognition.
I'm not complaining.
No, I don't think
I'm infamous enough.
All right, you're not complaining, but I think it's true. So the summation of this whole thing is that we're dealing with something which really is a clinical problem of very great importance, being done by people who have done basic science, very high level, very skeptical scientists, very critical, working on very hard problems, published, I don't know how many papers we published at this point, maybe several hundred, maybe 300. Had lots of good students, lots of good postdoctoral fellows, lots of good associates who've gone on to be professors or researchers at other places. And here we are working on a very mundane problem, which is the glutathione levels and how that affects HIV replication, but at the same time as being mundane, it isn't mundane at all. It's of crucial importance to the world. And actually, I think it's intellectually getting more and more interesting. The article in the New York Times that you talk about is something derived directly from the work that we've done. We've talked to Fauci a lot. I've also talked to Meister. He found out about our results, and he's now extended it, I think, very slightly, and used a different compound or a couple of compounds. And there it is written up in the New York Times, and the story is open. I think that's where we have to end it. It's an open story. Will we or won't we, by following more of this work, come up with something which will really be useful in treatment of AIDS and a variety of other diseases which, by our research and others, seem to be also treatable by watching out for glutathione levels and being able to give an acetylcysteine, a very safe drug, one that was out of patent 30 years ago. I mean, it was patented 30 years ago, so it's out of patent. And nobody's going to make an enormous amount of money out of it, except by, well, I guess, period.
Well, I guess I misspoke, really, when I say that this is going to be the last word. Maybe it's the last word on the AIDS, but I think one thing that I do want to talk about, because you extend your own kind of feelings toward yourself to the people in your laboratory, and maybe just a few words on your laboratory styles and how you deal with your associates, because I don't think it's probably the norm.
No, it's not a very common style. But there are others who follow a similar style or aspects of it. We have a large lab. We've always been together, and therefore we've been able to manage more people working with us. And we like people, and we like to work with a number of people. We've had a lot of students, a lot of postdocs, as I say, a lot of medical students who rotate through our labs. And a characteristic of our lab is we've always had on Thursday nights, usually on Thursday night or some other night of the week, a journal club, which has been going on.
We call it the oldest floating crap game at Stanford.
Yeah.
It's been going on since we came in 1959, and it's had in attendance some of the most famous immunologists in the world when they've been here on sabbatical or been here for short times. But it meets always in our living room, except for a few times when it floated around.
Well, when the group was smaller, I think this is important. When the group was smaller, we could meet in other people's houses. There would be enough room. Now the group is a little bit too large to conveniently meet in other people's houses. Also, the Bay Area has gotten so much more spread out that people live too far away from each other for us to be able to go to each other's homes that easily. A few of these environmental problems.
Doesn't float as easily, but it's still the oldest floating crap game at Stanford.
Yeah, so
that's crucial. It's one that's known for its style. It's been started up in a number of other places because it's a no-holds-barred examination of a paper. It's typical for a journal club, but the neat thing here is that people are informal, they're relaxed, they're sitting on the floors, they're eating pizza or whatever they want to bring along. But they really go in very carefully to analyzing the data, the methods, the conclusions of what's there, and try to find what's good as well as what's bad.
Well, it's an extension of what we learned at Pasteur Institute, where the informal getting together for lunch and talking about whatever topics we wanted to talk about, and sometimes organized topics, sometimes not, it was a very, very seminal experience, and we followed up on that. Other people have too. But I think what characterizes our lab is that we're very close to most of the people in the lab, and they're close to us, and we know what everybody is doing. We pay a lot of attention to what is going on. We've been described as running a tight ship, where we know each of words. In other labs, you have somebody working by himself, talks to just the professor, doesn't talk to the other people. People are very competitive between each other. They want to get the recommendations to go to another job somewhere. In our lab, we really require a lot of interaction between people. In addition to stimulating it and keeping track of what's going on ourselves, we tell people if they're interested in related areas to collaborate, to work together. We almost always have two people at least working on any project. We try to avoid anybody getting away with cheating in science, fraud, something which comes up about. We had a recent scare about that somebody might have been doing that, and it wasn't. But there were a lot of errors, and we got the errors corrected. So I think you could characterize our style as having a family in the laboratory, and we probably spent a lot of time, much more time with the laboratory people, both in science and socially, and talking about politics and other matters than the average lab.
Add a little bit to that.
Sure.
Probably because when we come home, our friends can be the people from the lab, because I'm not sitting at home or with a different career where we have to merge our social groups or have a separate social group from that. So our social group has always largely been within the lab. Later on, you'll be filming some of the other people here. Recognize that the students and the fellows tend to come and go, but the staff tends to be forever. And David Parks, I first met in 1969. He joined the lab somewhere around 73 or 75. Tom Nozaki joined the lab around 73. Wayne Moore joined the lab in 1970. All of these people persist from another era. They also persist from a time when we found it was very important to make jobs for people who were working in war industry and wanted to shift over. So now we're talking about the 1968 to 70 time frame. A lot of the cell sorter owes a lot of its creative talent to people who just simply didn't want to work in the war anymore, and we were in a position to set up some jobs and do this, and it was consistent with our social and our political philosophy to get the best talent to work on the scientific projects that we wanted. But people joined the lab and stay because we try to keep, it's kind of a family, a humane environment, if you like. That means that everybody is allowed to work at least 20 hours a day instead of the usual 8. I mean, that's almost the definition of it. We're all around the lab. On the weekends, you'll find the students in the lab. That's not true throughout the building, but it's true in a number of labs, and particularly in our lab, that there'll be somebody there all the time. Our own family, the children, have all spent at least some time working in the lab over their period of schooling, and they